Yes.
Okay. A very good morning, everybody, and welcome you all to the 18th webinar of Plant Breeding webinar series. These webinars are being organized by the Foundation for Advanced Training in Plant Breeding on a monthly basis. And today we have Dr. Ramanatha Rao, who will talk about cultivation of tropical fruit genetic resources with a focus on utilization. The purpose of these webinars are to sensitize people to know and value the importance of plant breeding and also to create an awareness on various aspects like technology integration, policy supports, regulatory overview, farmers' comprehensions, and various other aspects required for inclusive development of agriculture. Foundation for Advanced Training in Plant Breeding, abbreviated as ATPBR, is a non-profit organization which aims to promote advanced technology in plant breeding and develop qualified plant breeders who would complement and add value to the existing system. ATPBR organizes professional trainings, workshops, and various awareness sessions to build required competencies of the global standard. And we also organize annual conference named as Fiber Farming Conference in different locations across India. Now, I would like to take a privilege to introduce our moderator, Dr. M.J.V. Rao, sir, who is a co-founder of GRSV Consulting Services, Bangalore. He has 1.5 decades of plant breeding related work experience in public sector, including US Bangalore, ICR, and ICRISAT. Later, close to 2 decades, he worked with Adventa India in various capacities and rose to position as a senior VP heading. Afterward, he moved to Metahelix Life Science Limited as President Agriculture Technology for close to a decade. He contributed for the development of new varieties and hybrid and published books and more than 100 research papers. Recently, he has been awarded with ATPBR for his Lifetime Achievement Award in the area of plant breeding. And it's really privileged to have you with us today. Welcome to you, Rao, sir, and over to you.
Yeah, thank you, Dr. Aparna, for that introduction about me. And thank you for giving me this opportunity to. Am I audible?
Yes, sir.
Okay, thank you.
Yes,
I
loud
have
and
great
clear.
pleasure. Sorry?
You are loud and clear, I said.
Okay. I have great pleasure in introducing Dr. Uttarapalli Ramanatha Rao. He hails from Andhra Pradesh, but then the surname in his name, Uttarapalli, actually represents Uttarhalli of Bangalore. So he is currently a Bangalorean and a person from Karnataka. He obtained his doctorate in genetics and plant breeding from IARI in 1975. In 1976, he joined ICRISAT at Hyderabad and to work on genetic resources of groundnut. And he worked at ICRISAT till 1989, almost for 14 years. During that period, he helped establish the groundnut germplasm world collection, numbering over 10,000 accessions. And I remember seeing him traveling all over the world where groundnuts are grown, collecting them under extremely difficult conditions, including several wild relatives. He worked very closely with groundnut improvement scientists, especially breeders, and I was a recipient of his good work of collecting the germplasm. And he participated in studying various trait development programs and the breeding efforts. After leaving ICRISAT, he joined the International Board for Plant Genetic Resources, which became International Institute for Plant Genetic Resources, IPGRI, and later Bioversity International. He initially worked at Rome, Italy, and shifted to Singapore, and later at Kuala Lumpur, Malaysia. His work has ranged widely and it has covered almost every aspect of plant genetic resources management, both in India as well as globally. And the aspects that he has worked include genetic diversity studies, utilization of germplasm, ex situ and in situ conservation strategies and practices, policies and programs plant genetic resources networks and collaboration, and the science part of genetic resources conservation. He has been a leader in his own perspective and has been a great team worker. One thing that, in the last 50 years, while germplasm was considered as a, heritage of the humankind, global heritage, from that period from that kind of thinking about 50 to 60 years back, now it is considered as a national sovereign, endowment or a resource and a right. And I think Dr. Rao has seen all that changes and helped a number of countries develop their germplasm policies and programs. He's also he has conducted several international training programs and developed many future next generation germplasm resources people. He has he's an expert in developing proposals on for utilization conservation of germplasm, which has received significant funding from various donors. And if I understand correctly, one of the latest work that he has done is on conservation and sustainable use of cultivated and wild tropical fruit diversity. And in a way, today's topic of his talk relates to tropical fruits diversity. After 2008, he has been an honorary research fellow with Bioversity International, and he has done a lot, continued to work on germplasm diversity and resources during that period. And he is currently one of the co-founders of GRSV Consulting Services, in which he and the chief editor have been bringing out a monthly agri-tech newsletter from GRSV. I think now getting into the topic a little, I think we have seen in India a green revolution from 1950 to even now. The cereals and field crop production, the so-called food production, has grown up from about 50 million tons in 1950 to over 330 million tons. But parallelly, very little spoken is about the fruit and vegetable production, which also grew from about 30 million tons in 1950 to over 340 million tons in 2020. And among the fruits, the focus has been on mango, banana, citrus, guava, and apple, which occupy almost 72% of area. But related to today's topic of which Dr. Rao will be talking about, there are a lot of unexplored and underutilized fruits that are becoming popular, and they're coming out from the genetic resources. Like we have not seen dragon fruits. It is now available in the market, which is locally produced. Trees like avocado, and there are several others which have a potential to become a provider of nutrients. And we are all very eager to hear Dr. Ramanatha Rao's expertise on underutilized tropical fruits. Now, before I hand him over the stage to talk, I have a couple of home rules before I hand it over to him. Because ATPBR is interested in developing the future generation of plant breeders. Well, we have a number of young breeders here, and we want them to ask questions. So please do ask questions without any hesitation. Prefer if you could type it out in the chat box, which we will organize and then present it to Dr. Rao for him to answer, or just raise your hand, and time permitting, it will be taken up at the end, and you'll get a chance to ask him the question directly. Over to you, Dr. Rao. Thank you.
Thank you, Dr. Vasudeva Rao. That sounds a little bit formal. Thank you, Vasu. As Vasudeva Rao just mentioned, I had definitely training and in plant breeding and genetics and plant breeding, but however, I have now been really a breeder. I wanted to make that clear to this audience. Although at this point of time, audience seems to be most of us that are visible on the screen. And second, most of you know me pretty well. And secondly, I have now been a horticulture specialist. Whatever horticulture I have learned is learned on the job. Basically, I am being plant genetic resources person with lots of interest and work in biodiversity all over Asia, I would say. And today's, as mentioned by Vasu, we will be mainly talking about tropical fruits, and in particular, tropical tree fruits. Because if you look at now the differences between the tree fruits and other kinds of shrubs and other things, there is tremendous number of species are involved. It's not like talking about either rice or wheat or maize, single crops. We are talking here a group of crops, going to talk about them, and certain generalizations will have to be made. But one, we must realize that everything we have to do is context dependent and crop dependent. And as mentioned by also earlier, there are so many underutilized species which are waiting to be exploited by humans. And there is hardly any genetic information available on them. So there is a huge gap there. And whether anytime anyone will be working on them is not very sure at this point of time. But we will see a little bit of all of them as we go along. And now I am trying to go to the entire screen. What do I do here? Aparna just told me, but it looks...
Yeah, so at the bottom you see there's one arrow.
Yes, I see. I click that.
Yeah. So then you click on entire screen.
Click on that. Yeah.
Are you?
Yes.
Yeah.
Yes.
Yeah.
Now you open your
present.
presentation.
Yeah, it's coming.
Okay, you can see me, but I can't see you. So
make it full screen, please.
yeah, I'm. So this is the topic. I just like to say again, tree genetic resources of tropical fruits. That's going to be our focus. And in the introduction, I would like to say that I'm sure that there are quite a few horticulture people here, like Sharon, Tiku, and all that, who know that horticultural crops have been growing faster than any other crops, even put together, for example, cereals. And hence, the genetic resources of tropical fruits, especially of trees, is also increasing tremendously. There have been many efforts and are ongoing efforts of conserving many of these horticultural species in the different institutions world over. But there are several drawbacks in them which we will touch upon as we go along. And as I just mentioned, horticultural genetic resources comprises cultivated species, wild relatives of several horticultural crops like fruits, which include trees, shrubs, tropical, temperate, etc. Each of them having their own requirements for conservation, vegetables, medicinal plants, spices, etc. And also the what are generally considered as plantation crops. And they require, if one has to cover, it will require many different and many talks over a period of time. Here we focus on a small subsection of it, that is tropical fruits. Conservation of species and genetic diversity, we'll try to take two of them together. And here is what we mentioned just now as the putting these all together, different crop groups and different species groups as the plant genetic resources. And worldwide, as I said, just mentioned, collection and conservation efforts have been going on. In India, these efforts actually started way back in 1820, when under the Raj government, they started the Agri-Horticultural Society. And presently, most of the work is being carried out by under the guidance of National Bureau of Plant Genetic Resources and by various horticultural and research institutions. For example, we have Indian Institute of Horticultural Research raised here, here, and plantation crops and so many other things, institutions. Almost all efforts to date have focused on ex situ conservation. I would like to emphasize that, with a very few exceptions, like in situ gene sanctuary that was established for one citrus fruit that in Garo Hills of Meghalaya. Even that, what is the present condition, we are not really aware. It has been done quite long ago, but not many people have have shown interest in going there and studying those species there. So we need increased efforts on conserving in situ, because many of the tree species especially occur in their not really not just wild relatives, but also their wild forms in many forest areas, some of which are protected, some aren't, and such things need to be protected for future, and we will see why that is very important in the case of fruit species, especially tree species. Collecting and conservation of only of tropical fruit trees only started in 1990, I would say. I mean, as I mentioned earlier, people have been collecting them earlier, even from long time, especially in other countries. But the concept of collecting genetic diversity was only came into picture in 1990. Before that, it was mainly collecting what horticulturists referred as allied trees. There was never, like rice or wheat or other cereal crops or legumes or even shrub species. Collecting of genetic diversity, a population collection. So only in 1990, we tried to change it a little bit. Even today, it's not very successful, but relative to 1990, it's much better situation we are in. I will come to that little later, what exactly these differences are. And given the problems of various the fruit trees, the perenniality, long gestation period, a tremendous amount of land that will be required. Let us say, now you can evaluate, 1,000 groundnut varieties, in an acre of field. But whereas you can do the same thing with mango, for example, which has got over 700 different varieties of Mangifera indica, let alone other mango species which are consumed in Southeastern Asia. So characterization and utilization have received much less attention in that sense, relative to other cross species, due to the inherent problems of that's what we just mentioned. But that's all because of the inherent problems of tree species. Not just fruits, for example, any tree species, including forest trees, this has been fairly slow. So, I don't know whether I had to go into the basics of conservation at this point with this group or not, but basically what is done in collecting is people go out into the fields or into the forests, try to collect samples of different plant species in which they are interested, bring them back and try to conserve them, mostly either in seed banks or gene field banks or in vitro collections, or even you can freeze dry them and store them at -196, actually -176 or so, in using the liquid nitrogen. So these are various methods that are applied, details of which you can find in any of the basic plant genetic resources book. So as I mentioned earlier, most of the time people have been collecting, the allied materials, hence the collecting one or two allied materials from a big population doesn't represent the genetic diversity that is required for use in the future. Especially imagine if you would want to characterize and evaluate a group, a population, and select that is suitable for your purposes. Collecting, clonal materials is of very little consequence in terms of population genetics. But however, people have done tremendous amount of work even with that, and imagine what they could do if they have collected larger amounts of diversity. So there are problems with methods of collecting and transfer to gene bank, etc. Because especially tree species, most of them, especially fruit species, most of them are what we call recalcitrant. That is, you cannot really collect seeds, dry them up, and germinate them after 2 months or a year or so, because most of them are what we call they are recalcitrant, and they only germinate as long as moisture percentage in the seed is above 14 or so. Some of them even require up to 22% moisture in them for proper germination. So the advantage that cereals, legumes, and other crops have, seed crops have, the most fruit trees don't have. A few of them have, but not many. And also the size of the seeds sometimes can be prohibitive, like jackfruit and other things. And so sometimes that can be a problem even collecting. And for most species, appropriate methods of in vitro collecting are yet to be developed, because you can you collect tissue and even bring it back to the lab, regenerate, and either grow them in the field later, or even conserve them in what we call in vitro gene banks. But for many of these species, as I said earlier, for those underutilized species for which no protocols are yet available, it becomes very tough in doing so. These are some of the challenges that need to be resolved. And because imagine a cross species like mango, you want to conserve 100 accessions of let us say 40 trees each, just calculate how much land it will require. And land is a very precious thing, and it's going to be pretty difficult. It will be possible only for huge institutions with lots of budget. And also it will be very difficult to maintain, because it is not going to yield that you can sell and make a profit out of it. It will be for conservation purposes, where the costs are involved, and sometimes costs can be very high, and they can be very limiting in maintaining large number of populations and species, or even accessions. So because of this, limited genetic diversity of many of the fruit species has been conserved. But still, as I said, the characterization has not been very complete in many cases. There is a lot of work that needs to be done in the area of characterization, evaluation, and documentation. Good examples are available for some of the major fruit species, which can be used for other fruit species as well. And another is the long gestation period. So it takes quite a bit of time from planting to flowering to the production of seeds, or even getting a good clonal material out of it. Even if you get clonal material, at the end of the day, you are looking for fruits and seeds. So it can be very difficult in managing the resource collection, let alone utilizing them. We'll come to that part a little later. So some of the modern methods like marker-assisted genetic transformation, gene editing, etc., have been available for some species like grapes, citrus, apple, papaya, and other many vegetable species. Actually, in case of tomato and citrus, some of them have been going on for last 2 decades. They have been in the markets in some countries as well, but they are not generally available for hardwooded species, tropical species like mango, etc., because of the problems of producing tissue that generates good well and becomes a plant later on. People are trying to overcome these problems. You can refer these 2 very good ones, latest articles that can be used for getting more information on these matters. And only yesterday or day before, I was reading an article on production of a good regenerative protocol for Democarpus longum. You may not be knowing this species, but it is an important but threatened fruit species, tropical fruit species from some Southeast Asia, and very well liked fruit and nutritively pretty high. People have tried to grow longan in the Kerala and the high rainfall zones, but not been very successful to date. And hence, because of these challenges that have been listed here, conservation of a larger pool of species and genetic diversity of tropical fruit species it requires much more effort in terms of human resources as well as financial resources. And similar things apply for characterization and evaluation. As you mentioned, for several of the minor or underutilized species, no methodology, standardized methodology is really available. People have been working, but in a kind of their own little world and without much input from outside. Genetic enhancement and access. Most of the germplasm, especially the wild ones, require certain amount of what we call pre-breeding or genetic enhancement before actually the breeders can make use of them. So that becomes a kind of either joint effort or effort by germplasm curators to enhance the material so that the material that they conserve is actually usable by the breeders. Breeders are already, in trouble with doing what they want to do, and if we put the genetic enhancement also on their shoulders, it will be very tough for them. And any enhanced material cannot just stay in the genetic resources lab or in the field. Breeders have to have access to it. And we also need better and faster crop improvement methods like we just mentioned above, because the long gestation period that tropical fruit trees require becomes almost prohibitive in terms of doing crossing and testing and releasing any usable varieties. People have been doing it, but not with much testing, and that's one of the reasons why many of the tropical fruit trees have failed, because they haven't been properly evaluated out in the field or in the farmers' fields, etc. So modern methods can help in reducing. We cannot have definitely the speed breeding methods that are applied in cereals and others, but you can really speed up if newer methods become available to plant breeders for utilizing the genetic resources better. So these are some of the examples of species that we are concerned here with, like mango, jackfruit, mangosteen, the avocado, the rambutan. These are just a few of them. There are about more than 25 to 30 important tropical fruits in India. Not all of them trees, and Southeast Asia and Americas have tremendous number. South America has, for example, 400 usable tropical fruit tree species, apart from non-trees such as passiflora, etc. So you can imagine the amount of work that requires, and also the human resources. And continue with the challenges, and we only mentioned earlier challenges. We tried here to provide certain answers to those challenges. One is the lack of basic expertise in botany and taxonomy. I am sure most of you are aware that especially in India, if you go to the biology education, there's hardly any student who take interest in skills of taxonomy and descriptive botany, which is very important if one wants to study biodiversity and wild relatives, without which you only doing mangifera spa. So you can't go beyond that, and you cannot really make use of you won't even know what material has been collected, etc. And very poor representation of horticultural wild relatives in many collections. For example, there are over 700 2,000 varieties of mango, and of course, India doesn't have too many wild relatives except Mangifera and Manica and some occasional other species like Vodarata. But even those numbers represented in the collections are very few. And you go to Southeast Asia, where all the other wild relatives of mango occur, including some of the wild relatives which are consumed, if not cultivated, but collected and consumed, like Mangifera custodi, Mangifera odorata, Mangifera odoraceae system, etc., are in the collections, or even in the distribution maps. Their information on them is very, very minimal. For example, this is the area where many Mangifera species occur. I don't know whether you can see the arrow on the distribution map here, but most of them again are known as Mangifera sp, because people who have done the survey don't know the taxonomy to actually identify them to the species level. But in any case, some of them have been very good, and a number of trees themselves have been identified to be conserved, and they have been protected by local people in many areas there. Difficulties in accessing and using geographic distribution. I gave an example now, and that's definitely the newer versions of GPS and GIS systems are helping greatly in this matter. But again, if you don't have taxonomic information, it's going to be pretty tough to deal with any of these issues. And gaps in collections, we hardly know. There are, I'm sure, even major established cereals and legumes, there are several gaps, because I have worked on them as well. But it's not very easy, because most of the time, if you want to identify a gap in the collection, you have to have two basic things. That is what we call passport information. That means where it has come from, where it occurs, what are the map coordinates for it, altitude for it, etc., so that it can be mapped, and other similar location can be identified and later on surveyed for collecting. Or you have to have full characterization evaluation information, so that you can identify what are the gaps in the genetic diversity that has already been assembled in a collection, and then look for that genetic diversity, or even simply put, the traits that are missing in a collection. So these two require, and with, I mean, I'm sure many of you, some of you who know this agree that very little has been done in this way. And several times you have to, you know, when someone is going on collecting mission or exploration mission, yes, objectives are set at the beginning of it. Some of the funding agencies are pretty strict about following the objectives as set, but we have seen that over the years, these objectives have to be revised as one goes along, and in many cases, because of the poor information, you might go to some area where either flowering and fruiting are over or are yet to start. At least you had to make note of what we call again the passport information, and any communities that are utilizing or involved in managing those resources, etc. So that kind of information will be very useful for in situ collection later on. How is it going so far?
Pretty good.
It's good, sir.
Okay.
It's going well, Ramnath. Keep going.
Because I can't see any faces, so it's
No,
kind
no.
of monological.
Perfectly fine. Perfectly
Yeah,
fine.
perfectly fine.
Thank
We
you.
just cut off the video so that the voice, etc., is very clear.
Yeah. That's normally what is done in a webinar anyway. Okay, thank you.
Yeah, continue.
So
I
just-
listening to you.
yes, please.
Keep going, sir. We can
Yeah.
hear you.
Yeah. Earlier I mentioned, there are different collection methods, and one of them is, actually, I consumed pretty good amount of time. Ex situ, that is outside its natural habitat, that's in your gene banks, in your research institutions, etc. These are all simple methods which are for which the guidebooks are available, especially if you're dealing with seeds. It's not big an issue, but in most cases, in our case, we will be dealing with cryobanks or even DNA banks in the future, and to some extent pollen banks, which are mainly used for conserving pollen to tide over the seasonal imbalances that may occur because of the different flowering periods of either different species or even different accessions. So I'm sure quite a few of you are familiar with these methods. And then we come to the in situ conservation, which, even personally, I think is more important for conserving tropical fruit trees because of the inherent issues that are involved in terms of the size, long gestation period, and the amount of finances that are required to manage them well in an ex situ collection. Even though an ex situ collection can be a backup collection for the in situ conservation. In situ is, for me, the primary mode in which we can go ahead do the tropical fruit tree genetic resources conservation. And again, some of the challenges here involved is very little has been done to date. There's hardly, I gave you the example of the citrus sanctuary in Meghalaya, but for that, there are hardly any examples, even outside India, let alone. There are definitely the community managed sites in both Southeast Asia and South America, and even a little bit in India, especially if you go into the Tamil Nadu area in the Western Ghats. But there is really no organized effort. Because unless the conservation becomes gets attached to an institution, it's going to be very difficult for continuing it for a long time. So we need to enhance the actions that in terms of policy support, as well as research and training. Especially, I believe in training because we'll come to that when I talk about human dimension later on. And close involvement with the growers and communities. Everyone knows about it, talks about it, that communities need to be involved in situ conservation, but when it comes to the action on the ground, it's almost always top-down approach. I am the scientist, I am going to tell you, please follow me. So that kind of approach, we come to that, which actually hinders the whole process and nothing really works out later on. And this very much linked with the next one, that is the understanding the concerns of the people dependent on NTFPs. Nobody is advocating, I am not advocating conservation at the cost of people who depend on them. So, and quite a few examples in our own homeland are available where the forest dwellers and tribals have been actually evacuated from locations for saving certain animal and plant species, which is a very poor approach to the whole process, because very fast the whole thing melts down, and at the end of the day nothing will be left. And totally one has to avoid top-down approach, even if when some principles have to be taught, better taught when you try to bring it out as if they know about it already, so that they own that particular thing and follow it easily, than saying, oh, this fellow is coming from outside and telling us what to do. So one can easily avoid that by using appropriate methods. In situ conservation is not feasible in all situations. Let us be very clear about that. They are context specific, and it's possible only in some areas and some contexts. So we have to identify that and only do that in that area with those communities and for those species. So it's not a panacea. Like in nothing is panacea. So limited allocation of resources, that's the crunch everywhere. I don't want to go into those details, but I'm sure we are all aware of this financial issues involved. Constraints to all types of conservation. So we put together both in situ, ex situ here, and I don't know whether this picture is very clearly available, but it's basically links each level of collection from field gene banks to the in vitro gene banks to cryo gene banks, etc., and provides also different things that need to be done, while managing, while even setting up the protocols and also managing later on. So one of the things that we'll find in any gene bank, if you go, is the lack of interaction between gene banks and users. Gene banks are doing their own work, breeders are doing their own work, because both are busy in their worlds. So this needs to be overcome. The difficulties in using modern tools, especially tropical fruits, several of them are very difficult to deal with. Our methods are yet to available, and especially when a germplasm regeneration becomes in hardwoody perennials like mango, etc., it's going to be pretty tough to deal with. And so we can finally conclude that no single methods can be useful for conserving the most of the genetic diversity that we require for future, especially considering what is going to come in terms of climate change. We had to evolve a better strategy, what we've been calling as a systems approach, that will take care of almost in entirety all actions required, all human resources required, put together these things in using the available finances and try to deal with this situation as well as possible. So this is what we call complementary conservation, and so that there is really no clear-cut hard and fast division between in vitro, ex situ and in situ, but it's a kind of a spectrum from totally ex situ to totally in situ. And this simple box here provides the way how to deal with, how to go from one level to the next. And in any case, we need networking of all stakeholders at national and international level, and define the objectives and analyze the feasibility of each option. Then decision on conservation option is taken, and setting up the enabling environment, both at policy level, legal level, and funding level, and elaborate the strategy of action to the why and to the stakeholders, depending on where you are, and then the implement process. This actually, we wrote it up way back in 2005, but even today we find it very difficult where people are actually doing something what we wanted to do. And coming to utilization part, I will not be going into details, but touch upon a few of the things. As I mentioned earlier, it's pretty tough in case of tropical fruit trees because of the various inherent issues that are involved. And again, this particular picture here from the FAO office provides some kind of guidelines how each kind of conservation, plant breeding, and seed production and supply are interlinked and can be, further linked to different processes and to the farmers or the agro ecosystems, whichever level you would like to see later, how this can be linked. Again, that will require a little more time to actually go into it, but I am sure you will get access to all these things, and you can later on look at it. And one more point is for the people who are actually students and others who are listening to it, please you can always write to me if you have any issues regarding to plant generating resources of not just tropical fruits. You can always email me. I am sure the organizers will be providing my email address, and feel free to write. What at GRSV, what we do is the sharing of the knowledge. We are not going to take it with us. So we'll try and share as much as possible. And PGR that are available for crop improvements, it's generic statements again. There are over 7.4 million excisions available in 1750 gene banks around the globe. And out of it, 25-30% may be actually unique, the rest may be duplicates. And duplicates in genetic resources are not considered as a waste. It's a good safety backup. Certain amount of duplication becomes essential because the breeders in different countries are working on similar problems, and they have to have access to the similar germplasm. So certain amount of duplication will be coming there. Out of these 7.4 million, about 10% of wild crop relatives and about 11% of CGRs is in a CGR gene bank. And you can see in this, you can see the roots and tubers, which are also classified, some of the horticultural crops, nuts and fruits and berries, there's 6%, about 4%, vegetables make up about 7%. And the rest, of course, the cereals is the largest, followed by the legumes. And some of these crops, wild crops, etc., are also considered as in between crops. It can be plantation crops or spices and all that. What is missing here is the medicinal plants. Maybe they are included under others. So what are the global challenges? We have seen some of the challenges at the individual gene bank level, and But the global level, you will see that germplasm is hardly used. We have been talking about the importance of it and how useful it can be for the future, even present. But at the end of the day, if you work out your mathematics, your maths, and you'll find only less than 1% in most crops. Of course, wheat, maize, and especially the invention of modern tools, it is even coming down. Hopefully, it will turn out to be something similar what happened, when the molecular biology came into being, and the molecular biologists told that we don't require any germplasm, we can pick it up from a banana from the market and put the gene. But I think that was too much optimistic, and things have been proven to be different. And most breeders use only working collections. That is, we all breeders have our own collections from which we try to, because it's not because of they're lazy or anything, but it's difficult to either get them, especially after 1992, getting germplasm has become pretty difficult. And also, most of the institutions do not have the resources, either land or finances, to take care, unless it's at national level. Even in national level, people have problems. In case of TFTGR, there are additional problems. Almost all of them we have seen earlier, so we won't go into these details. So if you follow the timeline, this is what it will take, and even for simple crop improvements, 8 to 10 years, and you can add few more years if you are dealing with the tropical trees. So with the modern methods, which is using genetic engineering, gene editing, using CRISPR and molecular assisted things, this is how a picture will change a little bit, the amount of time it's taken. And I try to use this slide, especially because it only talks about mostly fruits. And as I mentioned earlier, some of the fruits like tomato, papaya, squash, it started way back, and some of them are available in the market. So this is slowly progressing. And as I mentioned, for further progress to be made, especially in tropical fruits, is regeneration technology needs to be perfected. People are trying to bypass that regeneration technology that is essential at this point of time, but even those results are yet to be actually checked and rechecked and see whether they can actually work in different labs, especially in labs which are not as developed as some of the advanced countries. So that gives you some idea of how things are progressing from way back in 1992 to now. So, but at the end of the day, with the challenges of global warming, climate change, erratic rainfalls, etc., we have set ourselves the task of making agricultural production a sustainable one.
Ramnath, 1 second.
Please read.
Hello?
Sudhakar Reddy, can you please stop presenting?
Hello? Okay.
Thank you. Go ahead, Ramnath.
No, Ramnath, Dr. Rao, you have to present. Somebody else got in somehow. So
Okay.
you
I'm
have to
continuing.
reshare, please, reshare.
Yeah.
Can you reshare your presentation?
Hello?
You have to
Yeah, go
reshare
ahead,
your
Ramnath.
presentation. We can't see your presentation.
You can't see?
We can't
have to
see
reshare.
your presentation. You again share. Stop presenting and again share.
Okay, sorry.
Click on your presentation. You will see stop presenting, stop, and then again do the.
Yeah, I'm doing that.
Yeah, because it has gone out.
Where is Resume your resume presentation.
Yeah, it's coming on now.
Yeah.
It's coming now.
I don't know how it happened. Mhm.
Is it visible now?
Yes.
Okay.
Yeah, make it full screen. Correct.
It is visible. Please make it full screen.
Yeah, it's clear.
Yeah, I'm just moving it forward.
Yeah.
Yeah, you saw this, right?
Yes.
Yes.
Yeah.
This was the
Okay.
last one.
So sustainability is the buzzword now. I mean, it's not really buzzword, it's a must. If
Yeah.
human race, I mean, that's my, again, personal view. If human race has to sustain itself into the future, it has sustainable production becomes absolutely essential. And this sustainable production is not in just agriculture, but in everything else we do. But let's confine to agriculture here. And this is a concept based on intergenerational equity. It's something to do with equality, justice for equality, and all that. However, production practice any production practice will consume certain level of resource. So the question is how well we manage these resources and how well we manage these waste products coming out of that, and still continue sustainable production. That becomes very, very important. It is an aspirational goal. It's not a goal that can be achieved easily or even really achieved, but we require to work continuously towards this. There is no option for us, as far as I can see, with what is going on around the world, especially, going into social issues like greed and all that. It becomes impossible not to look at it, to work continuously on these particular aspects. So better appreciate the ecosystem services which are provided by agricultural needs, especially, is very important. So what are those services? You require soils, water, fertilizers, or nutrition, let us say, greenhouse gases that are emitted, how to reduce them or even how to use them, pesticides, let's get rid of them as much as possible. Biodiversity needs to be conserved at the same time without expanding the land that has been blamed, and rightly so, for the increased agricultural production in many cases. And food safety and quality. If food is safe for longer period and the quality is maintained for longer period, there will be less and less wastage. That's the main thing behind it. There's globally 14 to 18% of the food is wasted. That means we need produce additions. Or not only that, the wastage adds to the pollution because much of it is not really composted or to reused in a different way. So these are the things which we require. These are the challenges as well as the efforts, continuous efforts that are needed for achieving this objective. And the current econometric methods that are available for measuring the growth and production and the development need to be revised drastically. Talking just about, the other day we had a blog, and talking just about GDP is not going to help anybody. It has to be Human Development Index, and that should be the ruling index we need to use for the time being. Get out of this GDP rigmarole. And we also need, for us, decision support tools in different ways. For example, which are the soils are healthy? We know how to improve soil health. how to use water economically and with little wastage, and how to make plants more efficient in what to use. These are the things that we need to be looking at in the future. Agricultural biodiversity, especially tropical fruit trees, help significantly in the ecosystem services. Compared to many other simple herbaceous species, tree species have got additional advantage of recycling, water holding, and coming to the recycling of gases, many other ones, many other ecosystem services that are where even all the forestries are involved that can be obtained from appropriate planting and management of many of the tropical fruit species. So they have also, apart from food production, they also have a major role to play in how we are going to deal with our ecosystems in the future. So how do you improve our using PGR? We have seen that less than 1% are used. And but as the literature at least tells us, for in the last few years, especially starting from 2018 or so, agriculture is a victim of climate change as well as it is the means of mitigating it. It is a victim of climate change because in many places, if you remove, of course, animal agriculture part of the equation, the agricultural production from soils and other means is much less the impact on climate, but still they contribute considerably for the pollution in terms of pesticides and fertilizers, and also the increase in cost of production, which is a main resource, because of which people are always looking for increasing productivity for unit area. It has been our axiom for several years, which requires different thinking now. Agriculture biodiversity, the biological basis of innovative and resilience. As I said, especially the characterization and evaluation, but for the major food crops, let us say 10 of them, is still very backward. It needs to be improved for the traits that we will be looking for in the next 10 to 15 years, so that the temperatures don't go beyond 1.5 degrees centigrade or result in pollution of all our water sources, result in very poor soil health because of the extensive use of chemicals. So some of the traits can be found in the genetic diversity that's available to us. Even if it's not in ex situ, there are usable material nearby in the wild areas. So we can, using this agriculture biodiversity, we can become more resilient and productive and sustainable systems in the future by control of pests and diseases, not without many chemicals, regulation of microclimate, decomposition of waste, and regulation of nutrient cycles and crop pollination, etc. Although people have shown that most of the pollinators, including the bees, are negatively impacted by various pesticides, there are still large groups of people which say it is not. So there is an amount of reluctance on the part of people to emphasize this fact too much, but if you look up meta-analysis that have been done, studying, putting together more than 300 to 500 studies of the impact of pesticides on pollinators, there is a tremendous decrease in number of pollinators in any given locality. So some of the opportunities that are available to us, or reintroduction of land resources held in gene banks. I remember way back when I used to work in ICUSAT, there were farmers who used to come and ask for the land races, so-called land races that they grew in their fields. But unfortunately, at the time, the distribution policy was not very conducive to that, because these people were not considered as bona fide users. Bona fide users were only breeders. But later on, this has changed, and I think the more and more, if people want, this can be done, and this is much easier done in case of networking of the people, custodian, what we call custodian farmers, who maintain tropical fruit genetic resources than in the material held in the gene banks, because most of the material held in the gene banks represent clonal materials and single cuttings and budwood cuttings and from single trees, not genetic diversity itself, in which help to form the landrace. The varietal crop diversification, varietal mixtures, if you are following the current literature, you will see that tremendous amount of work is coming back on crop diversification, varietal mixtures, etc. For example, when we used to collect in from farmers' fields, so we have seen from 18 to 28 different species grown in some small patch of land. Those things have disappeared slowly over the years, and now scientists are telling farmers that they have to go back to that system. And may not be 18 to 28, but at least a few of them. Contribute to a reduction in input use for production, reduction in input use. We may not go to what is called either zero farming or zero input farming or natural farming, but definitely there is a need for complete, total rethinking about the inputs that we are using and how to reduce most of these things. Formal crop breeding must continue for adaptive traits. Need to integrate modern knowledge with local and farmers' knowledge, and risk mitigation and avoidance effect of varietal and crop diversification. It needs to be, there needs a little more effort in actually putting figures on most of it. Even now, some of the things are coming back, based on studies done on labs and institutional fields, very little from farmers' fields, for example. This maintaining the high response diversity can facilitate post-disturbance recovery to compensate for the negative effect of climate change. There will be definite effect of climate change, but I think we can survive if we start practicing this right now, not later. I think I skipped this because it is the same thing repeated. So we know that utilization is the main driver for most plant genetic resources. We specifically talking about tropical fruit genetic resources, especially in situ, goes beyond utilizing. It is the survival of humankind that will determine how much of in situ conservation is practiced, not just TFTDR, but most of the genetic resources, especially wild genetic resources, which have a tremendous future in the, I believe so. They should become integral part to a larger conservation effort. They cannot be done piecemeal. They become part of the strategy, and the national strategy requires rethinking on several aspects of this. Using tropical fruit trees requires, as mentioned earlier, pre-breeding, germplasm enhancements, etc. But if modern methods come into use, so maybe some of these can be discarded, can be got over with without carrying out for at least some species. Cost-effective long-term conservation methods are required, and this can be achieved if you use what is called systems approach. Systems approach is a little bit difficult concept to understand, but once you understand, then you will see the logic behind it. It is something like using the earlier while planning your project formulation and all that you used to use for certain methodology, and this is for not just project planning but for implementation of the results of the project. A systems approach can be utilized. In agriculture, it's been used in various other production systems, but in agriculture, it's just making an entry. We published a paper about 3-4 years ago on this, but still it's in formative stages. Use of modern scientific methods and tools instead of arbitrary identification and delineation of in situ conservation sites. We require more on-the-ground information, establish benchmarks for different locations and for species that may be conserved in those locations, and then only proceed from there. It needs increased research on population genetics, molecular mapping, and impact of climate change. Let us not just focus everybody on CRISPR or other things. You need a much larger set of disciplines to deal with genetic resources. That's one of the reasons why most genetic resources specialists are they are not really specialists, but they have a generic understanding of agriculture and other systems, are not specializing in any one particular aspect, and that's required for proper genetic resources management. Do you want me to go through this?
Maybe quickly, because we have already gone well beyond time.
Is it the I am on time?
Yeah, we need to give some time for discussions as well.
Yeah, that's why I asked, because
human dimension.
I think climate change is everybody knows pretty well now.
Yeah.
Yeah, I will skip and go to human dimension.
Yeah.
Because I strongly feel this area has been neglected for especially when it comes to genetic resources conservation and use. This is been neglected for a long time, and as we have seen, the use, conservation and use requires different what you call skills and understandings and attitude of people, towards the whole process. It's not like, for example, not degrading or anything, but it's not like you got a specific goal of reaching a market, for example, using plant breeding product. So there is a definite goal set. Here, the goals are a little bit what you call, it's a kind of a moving feast. Things can change. For example, with the introduction of climate change business, the entire objective has changed. But most of the breeding objectives, like tolerance to drought, tolerance to this, tolerance to disease, they remain the same. But the genetic resources, which have all kinds of things, conserving and using them would change very differently. So, I mean, that's very simplistic way of putting. I know it, but that's easy to understand that way. So if you look at humans, the people in general, improved understanding of the farming community and not just farming community, sorry, it should be forest dwellers and tribal people and consumers of agricultural products is needed. There needs a balance between that. You cannot just focus on farming communities, profitability, but they should be acceptable to consumers so that actually they buy and the community benefits at the end of the day. All efforts are sustainable There is absolutely no exception to that rule in the future, and should be amenable change that may have made a technologies change over time. So you need to keep track of what technology is coming into the picture, especially in the conservation area, in terms of population and molecular genetics, and try to revise your own application of it into the systems process and change it accordingly. So that's the people in general. And from the scientific skills, you need, actually one thing you're missing in people in general is one of the important things is the public awareness. Slowly, there is this change in the attitude of training of especially youngsters. This is changing. There are more people are participating in conserving biodiversity, especially in terms of schools and colleges. So this is changing, but the change is not fast enough. The scientific and technical personnel and need competent scientists, that of course is there, but need to be in tune with the basic philosophy of conservation. Why do we conserve? Mostly we're conserving for the present use as well, but for use in the future, for the future generations. That's that means you've got to get rid of some of the greed that may be there within us. We are struggling here to keep the things the way they have been, at least to the extent possible, so that the future generations of humanity can continue with at least the level of at which we have been living. It should not be averse to field work. Of course, that's common for agricultural field work, not just field work. But in addition to that, there's an extensive requirement for going into the wild and getting materials and getting information. So should be able to should be prepared to do that. Be able to relate and work closely with farmers and indigenous communities. That means you require quite a bit of participatory research methods need to be used, and your working style should be cooperative and collaborative. We cannot work in isolation. That's definite. Policymakers, there's increased awareness, not just in saying, we are doing excellent work in biodiversity conservation, like many of our leaders mentioned, but actually facilitating that in terms of policy support, in terms of funding support, etc., specifically earmarked for that. We hear lots of greenwashing going on around the globe, including India, but greenwashing is greenwashing. Nothing is going to help to conserve and utilize the genetic biodiversity that we have. And collaboration among all actors. So collaboration is the key. Without cooperation, no genetic resources work will be possible. Yes, individual, what we call custodian farmers have been collecting and conserving them in their fields, but even they have obtained many of the materials in cooperation with other farmers. And this is great effort, and we badly need networking of those people so that they can further collaborate and continue to do the good work. So for us, what we call 3M approach, multidisciplinary, multi-institutional, multi-structural, it is an age-old approach, but it has been buried, but it needs to be, put back into action right now. So that's about it. And just to conclude, a few words. We know no country is self-sufficient. It has been very evident since several centuries. But now with biodiversity and plant genetic resources being a national heritage, is becoming pretty tough, even within the region. And with the changes to the Biodiversity Conservation Biodiversity Act, I'm not very sure. I'm pretty doubtful where we will be going in the future in the country, but that's a different story. We need to develop strategies for conservation of horticultural generic resources. That is, we need to fully support, participate, and implement various conventions, treaties, agreements. Southeast Asia, South America are very rich in tropical fruit trees and tree species as well as wild relatives. So they require special attention. The value may not be realized now, but their value will be realized in future, because as the climate tends to change, new actors come into play and new species will be taking over some of the area planted by the older ones. Like Vasu was giving some examples in the beginning, in the introduction. There will be new and maybe better quality produce in the future. Multi-dimensional problems, so we require a lot of effort, especially convincing and getting the stakeholders to act. That's an important effort that's required. Issues that need to be addressed mainly are collecting exploration in situ on farm, evaluation and utilization, and change in the mindset of people involved. I call it as mindset, and because without which very little can be achieved in the future. And sustainability should be our main driving force. Everything we do has to lead to sustainability, and sustainable development process is augmented with whatever we do. I think that's about it. Thank you.
Yep. Thank you very much, Dr. Ramanatha Rao. Much appreciate your very detailed presentation. And a few questions have come in the chat box, and I think 2 people have raised hands. So I think out of the questions in the chat box, there are 2 questions which appear directly connected to the topic of your presentation. So I will ask them first. One is from Vaibhav Bansod. He is asking you.
Say again.
The question is from Vaibhav Bansod.
Okay.
He is asking the question, how can we use underutilized fruit crops in fruit production?
Can you please stop sharing the presentation so that everybody is seen?
Yeah, I'm getting out of it.
Yeah. Thank you.
Yeah, I think the answer is in the question itself. Start producing. Vaibhav, the best way is, very detailed answer has to be given to that. If you really wish, I can send you email, because answering that, at one go here may not be very easy. But simply again, if you have an underutilized fruit species, whether it's the tree or whatever it is, it needs to be brought under cultivation first. And then to evaluate and compare it with other fruits that are already available in the market. And you need to establish whether these fruits will be liked by people, whether they would pay money to buy them. That's clearly most important one. Within the forested areas, forest people, fringe dwellers would always use those things. There's no question about that. But to bring it out and as a marketable product, you got to follow the usual steps of what people did in the domestication process in the beginning. Bring it under cultivation. Check, what kind of yields they provide. Before that, along with that, check the consumer preferences, if any, and then only, you can really go forward. That's a very simple answer for what you have asked.
Okay.
And there's another question from Mahesh Kulkarni. He's asking, a lot of research work is going on biotech, bioinformatics, and genomic aspects of tropical fruits. Lots of research paper available, but why no single variety is released by any institution by using this information?
It is a question to be answered by, let us say, people who are actually involved. But my simplistic answer from what I have read is that probably the one is only looking at what is happening in India because of the policy issues. I am sure Vasu can tell you for an hour or two on that. We don't have a policy established for genetically modified organisms, for example, first. And CRISPR, I am not sure any work is going on in India. Maybe in 1 or 2 labs have started. Again, my knowledge in this is, I tried recently asking several people about this, but not many have come forward to provide me with an answer. Yes, as I showed you in one of the slides, in papaya, tomato, and all that, genetically modified organisms have been produced, even in fruits. They are available in the market because of these countries have favorable policies for releasing them.
Okay.
You may add Vasu and et al.
Yeah, and others. There are questions from Sudhakar Reddy and Ramlinga Reddy. I will request Ramlinga Reddy to please ask his question.
Yeah, I have some comments. Do you hear me?
Yeah,
Dr.
please
Rao?
go ahead.
Yeah.
Yeah, it is a nice presentation and very exhaustive presentation. And definitely there are certain difficulties when you are handling the germplasm of the, especially tropical fruit species. So whoever is involved in this process, they should classify whether it is a high risk species or medium risk species or low risk species. Because why I said high risk species means because it requires certain kind of infrastructure facilities for the conservation or while evaluation. That is very critical, because if you think like a papaya or banana or avocado, or for example, I am giving some examples. So these are citrus. So there is a number of viruses and viroids are there, and also phytoplasmas are there. So it is very necessary to integrate the testing procedures for these complex organisms. That is a very, very critical. And also in germplasm exploration, so some of the virus pathologies should be associated preferably. And also it should be before conserving, whether in situ conservation or ex situ conservation, there is a requirement to test it, either virus indexing or virus testing, to ensure that is virus free, because the virus and viroids are most notorious because you are dealing with a perennial crop. Once they got into the system, so it is very difficult to eliminate, and also the losses will be colossal. So this is very critical when you think of any horticulture plant species, tropical horticultural plant species, which is known to harbor many viruses. One has to be careful. As far as possible, where there is in situ techniques are available, it should be utilized, coupled with virus indexing. That means in case of some cases, tissue culture propagation methods are available, but this should be integrated with virus indexing procedures. See, protocol, there should be, as Dr. Rao also told, there should be some kind of protocols to be established. That's the most important. Infrastructure facilities, that means because there are some viruses where the vectors are involved. If you don't grow them at initial stage, at the time of testing, under insect-proof facility, then you will be spreading a different strains to different plant species, because some of these fruit plant species viruses will also goes to agriculture crops. So where the risk is more, because this will act as a perennial source. what I mean to say, these precautions should be kept in mind while conserving this genetic plant species of the, especially the germplasm of the tropical fruit plant species. Because one is whether it is an indigenous collection or exotic collection. So indigenous collection, at least they should know what kind of viruses prevailing in the country should be taken into account. What are the problematic ones? If it is exotic, besides you should have a phytosanitary requirements also prevails, because that is a routing through proper institutional framework. That is the most important.
Yeah,
These are
thank
some
you.
of my comments and suggestions.
Thank you.
One should really, it is a tropical, the germplasm of the tropical horticultural crops is one of the weakest area, because not much exploitation is done, because there is a lot of wild species also there which can be used for cultivation purposes. We do not know, because some of them also may be near extinction. If you take some of the wild phonic species, which we have seen in the earlier during our childhood, they are no more, because very good, excellent source of fruits. It does not cost much. Cultivation also, because, but these are almost under extinction because during the forest depletion. So these are some of the critical comments and my views I expressed, especially when you are dealing with the conservation of the
Thank you.
germplasm of the tropical fruit plant species. Because that's why one has to identify the species,
Thank you.
whether it is a high risk species or moderate risk species or low risk species. Low risk species will have a minimal facilities, minimal infrastructure facilities are required. But when you are dealing with the high risk species, you must follow certain strategies. Thank you.
Thank you, Ramanatha for. I mean, it's definitely. I didn't go into the details. You know, it is part of the challenges that tropical fruit trees genetic resources has. Any fruit, any perennial species conservation is very important, from seeing that it is as free as much free with any disease, especially the viruses, as you mentioned. There's no question about that. And that this is one of the reasons for many of the fruit and vegetable species, the people have adopted to go for in situ, I mean, in vitro conservation. So when I mentioned systems approach to conservation, systems approach is what we call part of the mind mapping. It will take care of all the elements that are involved from different angles and try to deal with that. So we have to use that approach, not just diseases. There are, and another thing is we should not be using high risk, low risk here, because high risk, low risk for most of the plant people would mean in terms of their existence, whether they go extinct soon or later.
No, high risk in the sense means.
Yeah, I know, I understand you. I have no question.
Yeah,
what you have said, I understand that.
Yeah, that's why I think.
So question of how we put things is also important. So I mean, I'm fully in agreement with you. There's no question about that. But
can we move on to the
into all those details in
mainly two problems are there. One is either symptomless, how you handle with the symptomless plants. Other one is the vectors. Yeah, these two are critical problems.
I understand that, but you see, even many more things that are required, we are pretty poor. So let us go.
Can you please allow others also to raise the queries?
Yeah, I think
Sudhakar Reddy,
are you?
Yes, I'm engineer and farmer with 15 years of horticulture experience. I notice the fundamentals are not being followed. In the simplest terms, before we even talk of conservation of germplasm, having understood the germplasm has been maintained. I've seen botanical garden in London and in also the Malaysia, the nursery, the one highest form, the other in a simple form. You see, they have been maintaining these nurseries. with in situ methods. So using marigold, the trap crops as pollinizers, and the pollinators are helping their own plants being raised there. And I've collected seeds of marigold from there. And then including from Hyderabad also, I have collected marigold. I want to see how they behave as pollinizers, to see how these pollinators will behave, see, in respective fields. I have 15 acres of mango in one area. About 2 kilometers away, I have another 15 acres of mango, where I always compare this microbiome between these 20-year-old trees and 10-year-old trees. My microbiome is neglected. You see, other day in PGPR international seminar also I told, unless you correlate the microbiome of the soil food web and the microbiome within the plant, like how you correlate with your own gut microbiome, these are all interrelated. Unfortunately, we have not gone to that level, although we are interdependent. I don't want to get into like classes here. I'm not a scientist. My simple question is, the fundamentals are known, principles are known. Practices should be converted in a simplistic manner, as to be convenient, either in terms of protecting it or promoting it. Like, for instance, if you're promoting, it should help you and help the other neighbor. And if you're not able to protect your crop, see, from this pest, say, as somebody rightly said, it would affect the neighboring fields also. So in that sense, you see, the conservation should have meaning as to really help the grower by following that conserved germplasm. So what we do is simplistic terms, as you rightly mentioned, we have to prune the crop. We don't do that, and we blame the species, and we don't lay the species, you see, one after the other in the rows. We all will put, you see, all the same type of species, you see, across the rows, instead of alternating these varieties, you see, so that the cross-pollination will have better effects, as you rightly mentioned in one of your slides. This is human intervention that is required, you see, and then the extension, and then the grower, the symbiotic relationship is not there. We only talk of symbiotic relationship within the plant. We don't have synergistic effort, you see, between these. And I was richly benefited with these methodical approaches by following simple principles of nature, and like alternate wetting and drying. Incidentally, you get so many rains. What do we do? For 40 days, we have rains. And we brought the vegetative matter and then dumped onto the soil, and then made it, see, the pulp in the sense, in the rotavators we use, converted it. I don't want to again get into the practices here. What do your species do to overcome these climatic changes happening beyond your control? So therefore, my philosophy is that conservation of germplasm only will help to some extent. We have got, I think, 60–70% of practices without relating to the germplasm. So therefore, my plea is we have to follow basic principles of approaches, practices, without regard to all that. And then you have to, microbiome should be given importance, especially by using proper biofertilizers in a methodical way. And my 80-year-old mango tree was full of mangoes when we started using these principles. I don't know what germplasm it was. And one can use those branches and then take it for grafting and use in all over Telangana. See, such things should be followed in stream. So therefore, we are not taking science to its logical conclusion. Thank you.
So I think Dr. Ramanatha Rao has a quick comment on what you have suggested.
Yeah, no, I mean, see, again, we are going into pretty details.
True.
This will be part of what we call how you deal with the agronomical aspects of field gene banks. Even way back in 2002, there is a training guide for field gene banks which you have prepared. If you look into that, these practices are listed. But many gene banks do not have the resources to follow them. So as you rightly said, this science is available, but practice, because of the lack of resources of many kinds, not just one, including human. That's why I mentioned human resources, how important they are. So it's a question of how much we can put in.
Okay. Can I
At
ask
this point of
Suren,
time, microbiome and all that are important, no doubt, but they are not going to change the basic genetic constitution of the plant we are trying to grow. We are not bothered about the yield aspect here. We are bothered about continued existence of the tree. So there are various, what do you call, thresholds to be set in and trade-offs to be made.
Yep.
That's
Suren,
all I can say at this point.
Thanks, Ramnath. Suren, you wanted to say something?
Ajit, it's a lot of time already. Probably, I just wanted to maybe one point. You mentioned, Dr. Rao, that the pre-breeding aspect knows that the utilization of the variability is the least, and I wanted to add to that that is the least. Even in the very crops that we have done a lot of interspecific breeding, the top 5 crops, including like tomato, the studies say that we have not even utilized 5% of the variability. And in fruit crops, probably not much of that variability has gone into breeding. So I think there needs to be a policy level decision on how to support that kind of breeding. And your germplasm conservation, obviously, I don't think there is enough attention given to it. On the human level, I think the botany students need to be having a course on food crop taxonomy, etc., so that there are skilled people available to get into this. Into this. Thank you. I mean, there's a lot. This is an excellent lecture. Thank you. It has made me think lots of other things. Thank you very much. I don't want to be making more time. Vasu, I think you probably want to round it.
Yeah.
Thanks.
Thank
Thank you,
you,
Dr.
Suren.
Thank you.
Yeah, Ramnath, you want to say anything?
No, I just I would like to thank him for that support, and I also would like to point out it is not even 5%. The estimates put the germplasm utilization at less than 1%.
Yeah, in fruit crops even less. Yes. But I'm talking tomato. They say 5%.
Yeah.
Okay. Thank you.
Well, one of the tasks I was supposed to do was to summarize Dr. Rao's presentation, but I think he has done it very well himself in the last slide. So I take it for that it has been done. Summarization has been done. And personally, I would like to thank Dr. Ramanatha Rao for accepting our invitation and then doing this presentation. Though it overshot a little, but the discussions were very interesting. And I would like to thank all the participants for their patient listening and participation in the discussions. I would like to now hand over to Aparna for her closing remarks. Aparna, are you there?
Thank you, sir. Thank you very much. Thank you, Ramanatha Rao, sir. It was very, new information for most of the people, because majority of the people work with vegetables or cereals. So knowing a bit more about trees, it's new information. So thank you very much for sparing your time for us. And thank you, Vasu, sir, for, waking up in the night and spoiling your sleep. So thank you very much for giving your time for moderating this session. And thank you all the participants for joining this session. And I'm very delighted to announce that next month we have a conference in Delhi. It's about the okra crop. Maybe Tiku sir can give a remark on it, and then we can close it.
Okay, I mean,
Can you please present this link of recording, please?
Yes, it will be presented for sure.
we have a global okra roundtable in IRI campus Delhi, October 10 to 12. Okra has become #1 vegetable crop in seed revenue now in India. And the problem is that we are still working on the same resistances by recombining the same things for the last, 4 decades. The resistances came in the 80s. So I thought that it's a time that globally people can get in, because outside India also now, many people are now getting interested in the nutrition of okra. Because of the pandemic, it has become even more. So the idea is that the think tank around the globe will come together. And so any of you interested in okra, please see the ATPBR link and register. It will be a nice conference. Thank you.
Thank you.
Thank you, sir. So next month we don't have the webinar, and we will see you again in November with another topic with other experts with all of you. Thank you very much. Have a great
Thank
day.
you, everybody.
Thank you.
Bye, everyone.
Dr. Rao.
Bye-bye.